All right, folks, listen up. If you're tired of confusing wellness advice and feel like you're missing a crucial piece of the metabolic puzzle, you're in the right place. Today, we're diving deep into a fundamental cellular process that very few people truly understand, yet it profoundly impacts metabolic health. I'm Lena Cruz, Metabolic Fact-Finder at Peptides.today, and we're about to unveil the intricate science of insulin folding. This isn't about willpower. It's about pure, unadulterated biology. Many focus on food, exercise, or blood sugar. However, deep inside cells, insulin folding is a precise biological process. This process fundamentally determines whether the body can produce working insulin molecules at all. Insulin folding happens long before insulin ever reaches the bloodstream, deep within cellular machinery. If this precise process goes wrong even slightly, the body may struggle to control blood sugar levels. In some research scenarios, errors in folding can lead to observed challenges early in life. Understanding insulin folding clarifies why genetics and intricate cell biology matter so much in studies. Insulin folding refers to how insulin is shaped inside pancreatic beta cells before it becomes active. Proteins like insulin must fold into precise three-dimensional structures to work correctly and efficiently. Before active insulin exists, the body first makes a precursor molecule called proinsulin. Proinsulin is produced in the endoplasmic reticulum of beta cells, where insulin folding begins. During this stage, the molecule must bend, twist, and connect in exact ways to become functional. If insulin folding fails, the insulin molecule cannot regulate blood sugar effectively in cellular models. Even worse, misfolded insulin can damage the beta cells that produce it, observed in lab studies. Over time, this damage may reduce insulin production altogether, impacting observed cellular functions. Proinsulin is a single long chain made of three parts: A chain, B chain, and C peptide. During insulin folding, A and B chains form special disulfide bonds, locking the active shape. The C peptide temporarily helps align these chains during folding research for proper structure. Now that we've swapped dogma for data and started to demystify the initial stages of insulin synthesis, let's zoom in further and see the machinery at work when things don't go as planned. It's truly fascinating how delicate these cellular processes are. For more deep dives into complex biological mechanisms and peptide research, make sure you're following Peptides.today on all platforms. We're dedicated to bringing you clarity, not confusion. Insulin folding is highly sensitive. Even a small genetic change can significantly disrupt the process. One example observed in research is mutant INS gene-induced diabetes of youth, often called MIDY. In MIDY, mutations in the INS gene alter proinsulin structure, causing unstable folding outcomes. As a result, proinsulin misfolds and accumulates inside the cell, observed in laboratory settings. These misfolded proteins place stress on the endoplasmic reticulum, a condition known as stress. When beta cells experience prolonged stress, they become damaged or may even undergo cell death. Over time, observed insulin production drops in affected cell lines, leading to diabetes models. It's a common belief that all diabetes stems from lifestyle choices. However, research shows this belief is incomplete. Some forms of diabetes are purely genetic, as insulin folding research actively demonstrates in labs. In conditions like MIDY, folding problems are present from birth, leading to early onset observations. This mechanism highlights the diverse causes researchers study at a fundamental cellular level. When insulin folding fails repeatedly, beta cells face constant pressure in experimental models. The cell tries to fix it, but prolonged stress eventually overwhelms these vital repair systems. Less insulin leads to higher blood sugar, which further stresses beta cells in ongoing studies. Understanding insulin folding offers valuable insights into diabetes development at a molecular level. Current research explores new strategies to reduce beta cell stress and improve folding efficiency. This knowledge may eventually help guide future therapy development to support proper insulin folding. Understanding your body at this profound cellular level isn't just scientific curiosity, it's a powerful step toward informed health decisions and a deeper respect for biology's complexity. We're constantly uncovering new insights here. Make sure to follow me, Lena Cruz, and Peptides.today for continued research breakdowns. Before we sign off, a quick legal disclaimer. This podcast is for informational purposes only and is not medical advice. All compounds discussed are for research use only and not for human consumption. Always consult a licensed healthcare provider for medical needs and treatment.